Hello everyone, I'm Dr. Deepika Malik. Today's topic of discussion is industrial production of aspartic acid. Aspartic acid was first discovered in 1827 by Auguste Arthur Plisson and isolated in 1868 from legumin in plant seeds and from asparagus juice. Aspartic acid is a 4-carbon amino acid that is used not only as a standalone food additive but also as a raw material for the food and pharmaceutical industries. It is conventionally produced in bioprocess utilizing immobilized enzyme-mediated or bacterial cell-mediated bioconversion. Notably, processes that make use of Corynebacterium species under growth-arrested conditions in a membrane reactor constitutes attractive options for the industrial-scale manufacture of this important amino acid. There is a growing demand for aspartate as it is a component of aspartame, an artificial sweetener, beside its use as a food additive and in pharmaceutical preparations. Now let us see the definition of aspartic acid. Aspartic acid, abbreviated as ASP or D, also known as aspartate, is an alpha amino acid obtained as a product of the hydrolysis of proteins. Aspartic acid is not an essential amino acid, which means that it can be synthesized from central metabolic pathway in humans, that is, they can synthesize it from oxaloacetic acid formed in the metabolism of carbohydrates and do not require dietary sources. There are two forms or enantiomers of aspartic acid, L and D form, as you can see in the figure. Enantiomers are chiral molecules that are mirror images to one another. Also, they are non-superimposable on each other. Of these two forms, only L-aspartic acid is directly incorporated in the proteins. The biological roles of its counterpart, D-aspartic acid, are more limited. Now let us see some physical properties of aspartic acid. They appear colorless, they form crystals, they are soluble in water and insoluble in ether. Aspartic acid is optically active, which means it is capable of rotating the plane of vibration of polarized light to the right or left. Now let us see some uses of aspartic acid. Aspartic acid is widely used as nutritional supplement and flavor enhancer in food production. Clinicians include aspartic acid in some natural programs for depression and immune function. Aspartic acid aids in energy production from carbohydrates and in RNA and DNA formation. Aspartic acid aids in liver detoxification from drugs and chemicals. Aspartic acid increases resistance to fatigue. As a dietary supplement, aspartate is combined with minerals like copper, iron, magnesium, manganese, potassium and zinc, and it increases the absorption of the minerals. Some forms of the supplements are used to reduce brain damage caused by cirrhosis of the liver, that is hepatic encephalopathy, when administered intravenously by a healthcare professional. L-aspartic acid is mainly prepared for the production of popular low-calorific sweetener aspartame. Now let us study the importance of aspartic acid. Aspartic acid is one of the two acidic amino acids, aspartic acid and glutamic acid. They play important roles as general acids in enzyme active centers as well as in maintaining the solubility and ionic character of proteins. Proteins in the serum are critical to maintaining the pH balance in the body. It is largely the charged amino acids that are involved in the buffering properties of proteins. Aspartic acid is alanine with one of the beta hydrogens replaced by a carboxylic acid group. The pKa of a beta carboxyl group of aspartic acid in a polypeptide is around 4. The pKa value is one method used to indicate the strength of an acid. The next slide shows the biosynthesis pathway of aspartic acid. It can be produced by two methods. The first is transamination of oxaloacetic acid. Aspartic acid is also an intermediate in the synthesis of lysine. As the glucose gets oxidized by glycolysis, phosphoenol pyruvate and pyruvate are formed. Both these metabolites can be converted to oxaloacetic acid, a key component of citric acid cycle. Aspartate is a product of transaminase activity which converts oxaloacetate to aspartate using glutamate as amine group donor. As you can see in the figure, the glucose obtained from the raw materials gets converted to phosphoenol pyruvate and finally to pyruvate during glycolysis. This pyruvate so formed gets converted into acetyl coenzyme A, which further enters into the TCA cycle. In this cycle, this acetyl coenzyme A gets converted into oxaloacetate, which acts as a precursor for aspartate. This oxaloacetate obtains the amine group from the glutamate formed by alpha-ketoglutarate and gets converted into aspartate. Another possible route of aspartic acid formation is by transamination of fumaric acid catalyzed by aspartase enzyme of bacteria. L-aspartic acid is produced using isolated enzymes rather than fermentation technique. The amino group is formed from the fumaric acid, which can be theoretically produced from glucose fermentation by the use of aspartate ammonia lyase or aspartase of E. coli, of E. fructans, and Pseudomonas fluorescens origin. This enzyme is quite unstable, but immobilization enhances its stability. An effective immobilized aspartate ammonia lyase or aspartase may be prepared by entrapping E. coli cells in kappa carrageenan. Once immobilized, the cells are quite stable, retaining aspartase activity for well over 600 days even at 37 degrees Celsius. The process is carried out in a packed bed reactor at pH 8.5 with the ammonium fumarate used as a substrate. Now let us have a look on the raw materials or substrates for the production of aspartic acid. The culture media are conventional and contains a carbon source, a nitrogen source, inorganic ions, and when required, minor organic nutrients. Suitable carbon sources include carbohydrates such as glucose and sucrose, alcohols such as ethanol, and organic acids such as acetic acid. As the nitrogen source, gaseous or aqueous ammonia, ammonium ions, and urea are preferably used. As the minor organic nutrients, preferably 2 to 8 microgram per liter of biotin is added to the culture medium. Especially in cases when the medium contains excessive amounts of biotin, the addition of antibiotics, surfactants, and antioxidants to the culture medium increases the yield of L-aspartic acid. Next is the selection of microorganisms. Several organisms are found to be suitable for the production of aspartic acid, such as Pseudomonas fluorescens, Pseudomonas aeruginosa, Bacillus subtilis, Bacillus megaterium, Proteus vulgaris, Escherichia coli, and Aerobacter aerogens. These microorganisms provide the aspartase enzyme, which is essential for the production of aspartic acid. Now next step is the fermentation. The traditional method of producing L-aspartic acid has been the enzymatic one of producing it from fumaric acid and ammonia or ammonium salt, utilizing the aspartase activity of microbes. Fermentation is carried out aerobically at 24 degrees Celsius to 37 degrees Celsius for 2 to 7 days. During the fermentation, the pH of the medium is adjusted to 5 to 9 with an organic or inorganic acid or alkali, or with urea, calcium carbonate, or gaseous ammonia. Now after fermentation, when the aspartic acid has formed, the next step is product recovery. First step includes the cell separation by centrifugation or filtration. Centrifugation is a common method used in industry and can be operated on semi-continuous or continuous basis. Poor centrifugation can be improved by adding flocculation agent. This agent will neutralize the anionic charges on the surface of microbial cells. Filtration is widely used in industry and can be improved by using filter aids. This separates unnecessary cell debris and impurities. Ion exchange is used for extraction and purification of amino acids from the fermentation broth on the basis of charge of the solution. Aspartic acid anions get bound to the resin. Next step is evaporation and crystallization. The aspartic acid solution is concentrated by heating, forming its crystals. The crystals produced are dried with hot air in a closed system. Then the crystals are packed in the packaging and ready to be sold. Another method for the product recovery includes precipitation at the isoelectric point. The isoelectric point is the pH at which a particular molecule carries no net electric charge. Isoelectronic point of aspartic acid is 2.8. Hydrochloric acid is added to bring the pH value of the solution to 2.8, which is the isoelectric point of aspartic acid. Aspartic acid is least soluble when the pH of the broth is at its isoelectric point. The solubility of aspartic acid being only 0.6% at this point leads to crystallization or precipitation of the greater part immediately from the solution. Now let us have a quick review of the process of formation of aspartic acid. The first part is media formulation with a suitable carbon source, a nitrogen source, inorganic ions, or sometimes minor organic nutrients. After that, there is selection of microorganisms. Then fermentation is carried out aerobically. After the fermentation is complete, next step is the removal of cells from the fermentation broth by separation techniques like centrifugation and filtration. After that, ion exchange is used for the extraction and purification of the amino acid. Further, the aspartic acid solution is concentrated by evaporation, which leads to the formation of aspartic acid crystals. The crystals obtained are finally dried and packaged, which are ready to be sold. With this, we wind up the topic of industrial production of aspartic acid. For any doubts and queries, you can contact me through the given email ID. Thank you.